Hello everyone. So today we are going to discuss about the cell wall synthesis, which is important in antibiotic, as we will see all the antibiotics how they prevent the cell wall synthesis. So before moving to the antibiotics and how they act, we have to study about the cell wall synthesis process in bacteria. This is a very quick video in which I'll tell about the few antibiotics and few drugs which prevent the cell wall synthesis, and in the next video I'll discuss about the beta-lactams and all the functions and how they act in a bacteria in very detailed manner. Okay, so first we'll discuss the cell wall synthesis. So this is a formation of cell wall. These are the various process. So basically we have three steps which are helpful in bacterial cell wall synthesis. First step happens in cytoplasm. This is a cytoplasm in which the first steps happen. So what is the first step? There is formation of building block. So these are some blocks which will form the bacterial cell wall later on. So first there is UDP NAC. This is NAC N-acetylglucosamine, which gets attached to UDP, and then it converts UDP NAM, that is N-acetylmuramic acid, with the help of enzyme enol pyruvate transferase. Okay, this is the enzyme which will help in formation of UDP NAM. After the formation of UDP NAM, there is a pentapeptide chain which gets attached to the UDP NAM. These final two amino acids are always DALA and DLA. These amino acids are important. We'll discuss later why the amino acids are important. These amino acids are formed when LALA is converted to DALA and DLA is formed, which is the last two protein of the peptide chain. This completed unit of UDP NAM pentapeptide is known as PARC nucleotide. This is known as PARC nucleotide, and it's combined with another NAC to form NAGNAM pentapeptide. Okay, so we have NAGNAM pentapeptide. We have the brick, which is important in formation of cell wall. So this brick is prepared. After the formation of NAGNAM pentapeptide in cytoplasm, it gets transported to the periplasmic space. So next step happens in periplasmic space. We have a lipid called bactoprenol. We have bactoprenol, which acts as a carrier carrying NAGNAM pentapeptide across the membrane in the periplasmic space. So as soon as it enters the periplasmic space, it will form a long chain, which is a polymerization step, and this polymerization step will happen with the help of enzyme transglucosylase. With the help of transglucosylase, there is formation of the polymerization. There is formation of long chain, which is a polymerization step. After that, we need cross-linking. Okay, as you can see, these are the cross-linking happening between all this NAGNAM polypeptide chain, and this is the last and final step. So what happens? How does this happen? This cross-linking happen with the help of enzyme transpeptidase, which is also known as penicillin binding protein PBP, which creates covalent bond between the peptide tails of parallel chains. So how does this happen? This enzyme, whatever this enzyme is, that is transpeptidase, it cuts off the last DALA. This DALA, which we have talked earlier, it will cut off the last DALA from the pentapeptide chain, and whatever the energy is released, the energy will help in forming bond. So DALA is helpful in forming bond after it is broken down by transpeptidase, and whatever energy is released, it will form the cross linkage. So this is the step of cell wall synthesis. Now we'll discuss what are the drugs which are present. First we have phosphomycin. This is the phosphomycin. if we'll talk about the drug, the first is phosphomycin. Phosphomycin, if we'll give phosphomycin, it will prevent this step, that is preventing enol pyruvate transferase, and the building block will not be formed. Second, if we'll give cycloserine, what will it do? It will prevent the formation of DALA and DLA. Okay. Next we have vancomycin. What will vancomycin do? Vancomycin will prevent the polymerization of the NAG-NAM polypeptide chain. So vancomycin will prevent this step. And then we have beta-lactam. What will beta-lactam do? This is the main hero, which we'll talk about in our next video about beta-lactam. So what beta-lactam do? It will prevent cross-linking by inhibiting the transpeptidase chain. So this is the whole process of formation of cell wall, and these are the various drugs which prevent the cell wall synthesis. Now we'll see how the mechanism of beta-lactam antibiotics happen. Mechanism of beta-lactam antibiotics. So beta-lactam, like penicillin, cephalosporin, are mostly bactericidal, meaning they kill the bacteria rather than just stopping its growth. And how does that happen? First, the beta-lactam ring, it looks chemically similar to DALA, that last two peptide we have talked now, that DALA. So beta-lactam looks chemically similar to DALA, and the trans peptidase enzyme. So where was the transpeptidase enzyme? This, if we'll look at the diagram, this is the transpeptidase enzyme. So what will happen after beta-lactam ring looks chemically similar to DALA, the transpeptidase enzyme mistakenly binds to the drug, and instead of the cell wall. So transpeptidase enzyme will bind to the drug instead of the cell wall. This will permanently inhibit the enzyme and inhibit the cross linking. So this is how the beta-lactam drugs will act. So in the next step, when the cell wall synthesis is prevented, when the cell wall synthesis is blocked, the bacteria will experience transcriptional de-repression. This will cause them to produce autolysin, which is peptidoglycan hydrolase. These are known as suicide enzyme, and they will start digesting the existing cell wall. So this is how the beta-lactam drugs act, and this is the step of cell wall synthesis and the various drugs which act via this pathway. So that's all for today, guys. In the next video we'll talk in detail about beta-lactams. For more such videos, kindly follow our channel, and you can also follow us on Instagram page. Our Instagram handle is Medicosphere. Thank you so much, guys, and follow for more such videos.